Thank you. Well, good morning, everybody. And I also want to express my gratitude that everybody has made the effort to be here in person for this meeting. It's fabulous to see faces that are so familiar that we haven't seen in so long, but it's even better to see all of the youth in the room because that is our future. And I thank you especially for being here. It's a great privilege to be the lead-off speaker for this event. So as Natalie mentioned, I have been working in mountain pine beetle by accident since about 2006. And I'm going to tell you a little bit of a different story today. It's a more recent project for us. It's only been 13 years in the making, this one. But first of all, I really need to acknowledge my collaborators and really the drivers of this project. We have Kathy and Rhiannon here with us today. They're going to rescue me if the questions get a little bit out of hand. They are the architects of this study, along with Josh Miller. But the genesis of this project is with an ecologist and entomologist, Staffan Lindgren, and I owe Staffan. Oh, and it says Staffan Lindgren there too, but that should say Dazine Huber. I owe them a debt of gratitude for launching this project, as well as Dave Coltman for all of his insights into population genetics to make this project a reality. So it's been more than 2 decades that Western North America has been victim to mountain pine beetle. This is a bark beetle that attacks the trunk of the tree. It carries a fungus that will grow into the sapwood, and proactively these trees are going to block water transport through triloses in an effort to block the movement of this fungus. And so that, in fact, is going to contribute to the demise of these trees. So 27 million hectares, conservatively speaking, have been hit by mountain pine beetle in those intervening 2 decades. So we're approaching the size of a good-sized European country, almost the size of Sweden. And this figure doesn't even take into account any of the damage that we would have seen in Mexico. So on the landscape, when we have large-scale epidemics where the populations get quite high and we have quite significant pine stocking, we can get these seas of red trees. And so many of you will be able to think back a couple of decades ago in the 2000s, where we had those pictures from British Columbia of those seas, those hectares of red trees. In Alberta, we don't have so many of these contiguous lodgepole pine forests, so it's a more patchy event. But when we get on the ground, we see these red needles. That's the result of that fungus growing into the sapwood, and especially at these high densities where we get these attacks very synchronously and over short periods of time, after mountain pine beetle has been through, we see miles upon miles of these dead trees. So even though those mountain pine beetle are voracious, even in these really extreme conditions, you can still find survivors on the landscape. So this tree, this picture on the right, this is taken on Norman Lake Road. It's about 40 kilometers from the house that I grew up in Prince George. And I took this picture. I know this picture was taken 13 years ago because I was pregnant with my son. And so Staffan took me out there and he says, do you think that genetically these survivors are any different? And the prevailing wisdom at the time was, well, these are just the understory trees. So they're not being sought out by mountain pine beetle. Maybe they're a little small, maybe they're kind of wimpy growers, or maybe they're just lucky. And I said, yeah, maybe, I don't know. We don't have the tools for this. Good luck with your project. And so Staffan didn't listen to me, because sometimes that's a good thing. And so he took advantage of the fact that lodgepole pine have serotinous cones. So even on those dead trees, a good proportion of the cones are not only retained on the trees, but they're retained closed because of the resin that seals those cones. waiting for a forest fire, because this is a region of the world where we get lots of forest fires. It's been in the news the last few years. And so those cones are closed, the seeds are retained, and so he could just go and take branches off of the survivors and the victims, extract those seeds, and grow up progeny from the victims and the survivors. So he did. And so he developed what we call the Lindgren collection. And so we have cones that were taken off of the victims. We know that those cones are produced pre-epidemic. We assume that both parents are victims because so many trees on the landscape were killed by mountain pine beetle. So we are calling these the susceptible phenotype. He also collected cones off of the survivors, and he collected them close to the ends of the branches. So we're assuming that these are produced post-epidemic. And so in this case, we can be fairly confident that both parents are living, they're both survivors. So we're calling this the resilient phenotype. I prefer the language of victims and survivors, but somehow it didn't sound right when we wrote the manuscript. So susceptible and resilient it is. So Stefan, being a great entomologist and a great ecologist, he collected from 4 different sites, including the Norman Lake site that I mentioned. And so we do have family structure in this collection of trees because only so many trees were collected from at each of these 4 localities, and multiple sites were collected at these localities. And so Stefan had these growing up in pots. They were in a greenhouse for, let's just call it, several years. And about 5 years ago, these were outplanted at 2 different research forests, one near Fort St. James for those that are from BC, and the other just east of Williams Lake. And for those of you that follow forest fires, yes, both of those regions took a big hit with the forest fires in the last few years. And we were lucky that Gavin Lake was protected by a fire break that ran through the larch trail that was right next to these. So both of these collections are still alive and well to the best of our knowledge. Let's see if they survive another forest fire year. So in order to address this question of whether or not there's a genetic component to survivorship, and from the title, you already kind of know that we did find that. We took what my postdoc advisor, John Davis, would call the triangulated approach. I'm calling it a layered approach here, where we're using a suite of different tools drawing from population genetics, quantitative genetics, and landscape genetics. So starting off with our population genetics, Rhiannon ran this beautiful minor allele frequency analysis showing that we do have a shift in that minor allele frequency distribution between the resilient and susceptible phenotypes, and this is significant at 3 of those 4 sites and quite pronounced at 2 of them. So there's a lot of interpretation that can come from these data. We're interpreting this as a genetic bottleneck from that extreme amount of mortality that occurred on the landscape. We also interpret this as reduced genetic diversity. So does that mean that going forward, we have reduced potential for that adaptive potential for other different traits? So that's a question, an open question, that we need to consider in the face of these kinds of extreme disturbances. But we also have this notion of this coevolutionary relationship between mountain pine beetle and its hosts. Through this selection pressure then, are we actually selecting for resilience to mountain pine beetle? Do those survivors have a genetic signature that is in common? So first thing that Josh set out to do when he went to address this question, he looked at heritability, and heritability was, holy cow, high. We were pretty excited by that. And then because he's a master at GWAS, he ran GWAS. Oops. And it was so darn disappointing that slide doesn't even want to come up. There we go. Missed it. There it is. Okay, yes, not even 1 point above the threshold. How many people have run GWAS and experienced this very same thing? Yeah, do you know, now we know we're amongst friends and we can share this kind of plot. That's all I got to say about that. But Josh had just gone to a genomic selection workshop. He was applying it in another project. So we said, well, let's just run that genomic selection. And now we were getting some promising results. So that was super cool. But the thing is about genomic selection, it's all great for breeding, but this was more a question about what's going on with that genetic signature. So we kind of wanted to know what were the genes of largest effect. So Josh ran this, what I think is a fabulous correlation analysis. And so what we saw is there is a great correlation between the genes of largest, or the probes of largest effect in genomic selection and the association study. And so he could use this to select those probes of largest effect, which we're now going to start to call genes of largest effect. We had a total of 210 of what we're calling genotype by phenotype loci. But we also know that we've got a pretty big geographic distribution between those sites. So enter Kathy's contribution to this great project. She does this amazing analytical complicated math in landscape genomics. And so she ran a couple of analyses to pull out 30 loci that we're calling our genotype by environment loci. 12 of those were in common between the genotype by phenotype and the genotype by environment analyses. And so we took those out of our candidate loci. And so this left us with just shy of 200 candidate resilience loci. So that is pretty cool. And what I did forget to say, and I apologize for that, is that all of this was made possible due to the grace of Sally Aitken and the Adaptree project by graciously sharing their AFI chip for us, because we probably would still be developing the resources to do this if it hadn't have been for that grace. And that's really important. I really want to underline in this talk that this project isn't made possible without the contributions of our forest genetics community. So thank you. So that's one population, but the next step that we need to do to see whether this pattern of being able to discriminate victims from survivors is to validate with independent genetic materials. So enter Nick Ukrainitz, who is currently growing up a number of families for us of, again, victims and survivor families that were selected out of their seed orchards when their seed orchards got hit a few years ago. So we will be continuing this grace of new funding that we have. And I'm really excited by this prospect of determining whether these models themselves are able to predict in this independent material, or whether in fact we could have to resort to a different model. But I'm a functional genomics person at heart, and I'm really interested in understanding what are those traits that are differentiating the victims and their survivors. So with this new funding that we've received, we have planning underway to go out and visit those plantations, start to take detailed phenotypic measures. We have a few ideas of what could be discriminating these two groups. Desine's group has already shown that terpenes, for example, do discriminate between families in subtle ways. So maybe there are defense mechanisms, but host quality in itself is quite a complex concept. So this isn't just about defense, it's about the whole of host quality. Through the annotation of those candidate resilience loci, we were able to find corresponding transcripts in our RNA-seq data sets that we have. And we find that there are 20 of these annotated candidate loci that correspond to transcripts that show differential expression in pines that have been challenged with the fungal associate, Grossmannia clavitra. This indicates that maybe these are genes that are involved in defense. So the undergraduates in the lab have been enjoying cloning those genes, and we've just started characterizing them, thinking that, you know, it's pretty much a needle in a haystack that any of these would be involved in that mechanism of resilience, but we have to start somewhere. And this is one place to start. And if it isn't part of that mechanism, it's another gene that's characterized in these massive genomes where we can't just rely on those angiosperm annotations to really know what these gene functions are. So with that, I need to acknowledge my whole lab, because at some point, every single one of them touches this project. There are a number of former people through the lab who have contributed directly to this project, but especially I want to thank the collaborators that we've had through this project, both those that were directly involved, those that facilitated, and those that lent their expertise where maybe ours was lacking to make this possible. And I did mention that we have new funding. This is through Genome Canada. Kathy is my co-lead on this. It's TRIA4. We have several postdoc positions open. We have student positions open. So if you're interested in joining the chaos that is this project, come and see me. Thank you.
Thank you, Janice. It was very interesting. We have 3 minutes for questions. I see Nana.
Okay. Yes, great talk.
Thank you.
How many surviving trees, or what was sort of like the frequency of survivors in an outbreak? What are we talking about?
So in the regions that were sampled, it ranged from 10 to 90% survivors. And that was at the time that that sampling took place. That mortality went up. So this is in a region of the province of BC where the ecologists call this the hyperepidemic. This was where the beetles were at such massive levels that they were flying vertically and then being carried by wind current because their behavior is changing. They were so dense. The populations were so dense that, in fact, they were attacking white spruce at some point. I mean, this is like Brussels sprouts to them, right? That's how many there were. You could even hear them under the bark. So in this case, that attack threshold was really high. And so mortality on the landscape was quite high. And these regions actually were stocked quite heavily in lodgepole pine. Lodgepole pine was major components of these forests.
So did you say 10% trees survive?
Yeah, it varied in which stand. So Staffen actually he did record that data, and it is actually in the Baylow paper, and it ranges from right across the board, but average was somewhere in the 50% range.
Well, and how many parent trees, or how many surviving trees in the resilient pool?
How many surviving trees were in the resilient pool that were collected? That's a good question, Rhiannon. Was it like 10, 20%? Yeah, that wasn't everybody who was in the stand, but I think he stopped at something like 20, and it was up to 20. In some cases, there weren't at 20 in that particular stand.
Okay, I think we gotta stop here. We will have the time for questions. We will have a panel later this morning. So we will we'll give you a chance to ask other new question to Janice. And thank you very much for your excellent presentation. Wow.